Ladies and gentlemen, welcome to our online meeting. My name is Dariusz Witt and I am a member of ProChimia Surfaces Evo Nano R&D team. And today I will talk about the convenient and versatile platform based on the functionalized gold nanoparticles for assessment of biological activity. Gold nanoparticles are available by different methods. Generally, they have used the same reagents and conditions. So it looks that the inorganic gold salt is reduced in the presence of stabilizing agents. As the most common salt is tetrafluoroauric acid used, stabilizers are carboxylate, phosphines, amines, thiols, or polymers. Reducing agent frequently is used citrate, ascorbic acid, or borohydrates. The gold nanoparticles are so convenient and versatile because we can control the size of these nanoparticles. So the size can be controlled by the rate of the reaction. If the reaction is faster, then gold nanoparticles are formed smaller. The amount of the stabilizing agent also is important. In the presence of the larger amounts of that stabilizing agent, smaller nanoparticles are obtained. Also, dispersion of the gold nanoparticles can be controlled at higher temperature. The monodispersed particles are obtained, and weak stabilizing agents also is responsible for the formation of the monodispersed nanoparticles. Also, the shape of the gold nanoparticles can be controlled, and as you can see, I have selected for you examples from the literature. And the most common nanoparticles are with the nanosphere shape. Also, nanorods can be obtained, and the bipyramids and the gold tetrahedra, octahedra, or cuba octahedra shape can be obtained. Also, nanocubes of the gold nanoparticles can be produced efficiently. In the case of the gold nanoparticles, we can also smoothly and easily control the properties of gold nanoparticles by appropriate functionalization of these nanoparticles. If the nanoparticles are functionalized with hydrophobic ligands, then the hydrophobic nanoparticles will be obtained. Hydrophilic ligands, they will improve solubility of these nanoparticles under aqueous conditions. So also important is that these nanoparticles are quite well stable in the wide range of the pH. Over here you can see example when the titration showed that the nanoparticles were stable between 5 and 11 pH. And what you can see over here at the right side are a solution of the gold nanoparticles, and when the pH is beyond the stability region, you can see that aggregation of the gold nanoparticles can be observed. Also, when we are going to modify the structure of the gold nanoparticles, we have to consider the introduction of the some compounds together with the background ligands. These background ligands we can use twiterionic thiols. Thiol group will be bounded to the surface of the gold nanoparticles. Also, bioresistance thiols with ethylene glycol units between carboxylic group can be used. And all these compounds are already commercially available from a ProChimia compound. And in the table you can see some examples which we have already prepared. When the gold nanoparticles with 5.5 nanometer size were functionalized with the appropriate carboxylic acid, this one, you can see that when the carboxylic group was converted into the salt, the measurement of the zeta potential reflecting the presence of the negatively charged functional group at the surface, and overall of the nanoparticles were with a negative charge. And when the appropriate ammonium terminated thiol was used for the functionalization of the gold nanoparticles, as you can see, the positively charged gold nanoparticles were obtained. In the presence of the positive and negative charge for the beta structure terminated tile, we have both charges. However, the overall charge of the all molecules is almost neutral. So as you can see, it looks that the positive and negative charge are in the same time will give you overall the no charges present in the gold nanoparticles. So how to introduce the active pharmaceutical ingredient into the gold nanoparticles? So as the example which I'm going to show you today, we have used 8-quinolinol, and we propose two ways for attachment of the API to the gold nanoparticles. These ligands were designed in such way that the tile group will use for the attachment of the all compound into the surface of the gold nanoparticles. The ethylene glycol units, they will improve the solubility in the water, and also they will provide the resistance of the gold nanoparticles for the non-specific interaction. And then, as you can see, the quinolinol is attached at the end. We selected two options. One, when the attachment is through the oxygen from the hydroxyl group of the quinolinol, and the second option when the API is attached to the additional amino group attached to the aromatic ring, so hydroxyl group and the heterocyclic ring could be exposed to the solution. The synthesis of these compounds was performed in the following way: that the hexaethylene glycol was first monoalkylated with the bromoundecene, and then followed by the free radical addition of the thioacetic acid to the double bond, and the protected thiol group was introduced in this way, and the methylation of the hydroxyl group at subsequent reaction with the quinolinol provided compound with the protected thiol group, and after removal of the acetyl group from the thiol group, the appropriate ligand was ready for the attachment to the gold nanoparticles. The structure of that compound was performed by proton NMR, for instance, where you can see the aromatic region and also the ethylene glycol functionality in the structure, alkyl hydrogens in the structure, and the most important, there is a signal from the methylene group directly attached to the thiol group over here. And in the similar way, the attachment through the nitrogen of the quinolinol was performed in a slightly different way, because the amino group was additionally present in the quinolinol. So we obtained appropriate active ester, which was obtained by the alkylation of the hexaethylene glycol, introduction of the appropriate ester into the structure, and then, as you can see, finally connection with the amino group which was present in the quinolinol. The final compound was of course characterized, and the proton NMR, as you can see, can be identified quite easily. We can see the aromatic region over there. Also, we can see the appropriate ethylene glycol, hexaethylene glycol units over there, and the methylene group close to the carboxyl group, and also the same way as it was previously observed, the methylene group attached directly to the thiol group over there. When you take a look for the theoretical calculations, you can see that the background ligand we selected a compound with the positive and negative charge, so the presence of the beta structure will improve overall the solubility of these gold nanoparticles under aqueous conditions, and also the appropriate length of the ligand with the quinolinol provided the availability of the API into the interaction, so it can be exposed into the solution. In the similar way, through the connection with the nitrogen of the quinolinol, again the background is shorter, so the active pharmaceutical ingredient can be exposed to the solution for the interaction. The preparation of the functionalized gold nanoparticles is quite convenient and straightforward. What you can see, the stabilized appropriate size gold nanoparticles. In our case, we have used 5.5 nanometer mixture of the tiles. One of them is the background tile, second is the tile with the API at the end. So the composition of the gold nanoparticles and the ratio between tiles attached to the surface in fact reflects the composition of the and the ratio of the tiles in the solution. So we obtained gold nanoparticles with 5.5 nanometers with the carboxybetaine background and the quinolinol connected through the oxygen in the ratio 1:1, and UVV spectrum simply reflects the plasma resonance of the metallic core, which is around 520 nanometers, and the DLS measurements showed the size of the nanoparticles about 13 nanometers. So it's larger than metallic core, but we shouldn't be surprised because DLS is able to detect and attach the ligands to the surface. That's why the overall the size of the nanoparticles is larger. And over here, TM images which are able to show just only metallic core of these nanoparticles. In the similar way, for the connection of the quinolinol for the nitrogen, we have obtained the size of the nanoparticles from DLS measurements about 11 nanometers, and the metallic core was from the TM measurements about 6.5 nanometers. It's average size. And in a similar way, we can see also the plasma resonance of the metallic core in the UV spectrum. In the case of the different ratio, we also obtained gold nanoparticles of the quinolinol connected through the oxygen with carboxybetaine as the tile backgrounds, but this time the amount of the quinolinol was larger, so it looks that the ratio between quinolinol and carboxybetaine is 4:1. And again, the size of the gold nanoparticles this time was from TM measurements about 5.2 nanometers, and the size obtained from the hydrodynamic measurements from DLS, it's about 10 nanometers, so definitely it's larger than metallic core, so it provides information that we have attached the ligands to the surface. And the ligand concentration was estimated about around 140 micromolar. General, the preparation of the gold nanoparticles with different size can be performed stepwise. So frequently the gold salt is reduced in the presence of the stabilizing agent. We use dodecylamine as the stabilizing agent, and in the presence of d-dodecyl dimethyl ammonium bromide and tetrabutyl ammonium borohydrate as the reducing agent, we obtained the gold nanoparticle sealed solutions, and these seeds were used for the further reduction of the net portion of the tetrachloroauric acid and in the presence of the stabilizing agents. And then you can see in this way we obtained 5.5 nanometers. The synthesis is performed stepwise because it provides in this way a much more monodispersed gold nanoparticles instead of the preparation in the one step. Also, these 2.5 nanometers gold nanoparticles can be used for the further as the growing seeds for the preparation of 12.5 nanometers. So as you can see, we can get about 2.5 nanometers, 5.5 nanometers, and 12.5 nanometers gold nanoparticles in the stepwise synthesis of these gold nanoparticles. And if the gold nanoparticles are introduced into the cell, it's difficult to observe them because they are too small, and frequently confocal microscope, for instance, is not able to observe them. So it's much better and convenient if these gold nanoparticles are functionalized with the tiles terminated with the fluorescent group, and the preparation of this functionalized with fluorescent dye nanoparticles can be performed in the same way as previously for introduction of the API. And we obtained in this way, over here there's just example, when the metallic core is with 2.5 nanometer and the background tile is the appropriate carboxylic acid with hexaethylene glycol unit between alkyl chain and carboxylic group. And as the fluorophore, we have used a CY5 dye, and we measure the size of these nanoparticles by means of DLS, and as you can see, the size is much more larger than metallic core. Definitely, it reflects the attachment of the ligands, fluorescent ligand, and the background tile. And then the zeta potential, because the carboxylic group is present at the sulfase, definitely will be in the form of the anion, so this negative charge is observed, and plasma resonance is typical for the metallic core. Also, we measure the fluorescence spectrum of these nanoparticles in the solution. The excitation wavelength was about 650 nanometers, and as you can see, emission was 672 nanometers. We estimated the concentration of the fluorophore as the 15 micromolar. To observe the interaction of these fluorescent gold nanoparticles with the cell, we selected a Melgiso cell line, and just to confirm that first of all the fluorescent properties of the gold nanoparticles can be preserved under physiological conditions. So over here the first photograph is just reference, second is after the addition of the gold nanoparticles, and what we can see, these nanoparticles are just too small to observe individual gold nanoparticles. However, what you can see that in the in the progress of the time, that these nanoparticles are gathering in some regions, and finally, after 21 hours, you can see clearly regions where these nanoparticles were aggregated, and the fluorescence of this region is very high. In the case of the similar experiment with the gold nanoparticles with 5.5 nanometers, you can see similar behavior, but the appropriate fluorescence activity is increased when the aggregation of these nanoparticles occurs in the cell, and then intensity of the region when these nanoparticles are gathered can be observed. However, in the case of the gold nanoparticles with the 12.5 nanometer size of the metallic gold, we have observed the quenching of the fluorescence activity of the CY5 dye, and we suspected that the fluorescent activity of these gold nanoparticles is quenched by the metallic gold. So we decided that instead of the hexaethylene glycol linker for attachment of the fluorescent dye, we decided to use a polyethylene glycol with the formula weight 3000. So in this way, the appropriate fluorescent dye is just moved away from the metallic core, so it can restore its fluorescent activity. And as you can see, it happened. So after 1 hour, fluorescence activity is pretty small, but however, after 21 hours, we have observed quite high fluorescence activity. So when the appropriate gold nanoparticles are functionalized, we also should consider that not only attachment of the appropriate compounds might be required, but the appropriate linker or the charge of the molecule must be also considered. So it looks that it's pretty complex, but we, in the case of the fluorescence gold nanoparticles, we can observe the behavior inside of the cell. At the end, I would like to just thank you, my collaborators, Agnieszka Lindstaedt, Justyna Doroszuk, Aneta Machnikowska, Alicja Dziadosz, Joanna Puzewicz, Piotr Barski. All these people contributed to our own results and the progress in the development of the research in the EvoNano project. And at the end, I'd like also to thank you for your attention.